Welcome to this AFOM Secret educational webinar series on the basics of nuclear medicine dosimetry. My name is Steffie and I am a medical physicist at the Radboud University Medical Center. And we have heard a lot already about the basics of dosimetry, and today I would like to tell you something about image-based post-therapy verification dosimetry. This is my disclosure of interest. So, as you already know by now, dosimetry can have a place in molecular radiotherapy during several stages of treatment. Before treatment, dosimetry based on a diagnostic imaging tracer can help in patient selection by providing insight in who is likely to respond to treatment. After each therapy cycle, dosimetry is often possible using gamma emission of the therapeutic tracer and can help to assess the absorbed dose to the tumors and organs at risk. By comparing this to the clinical signs and treatment response, we can derive absorbed dose effect relations, which in turn can help to optimize treatment schemes by making use of the therapeutic window, in which we achieve a high as possible lesion absorbed dose while keeping the absorbed dose to organs at risk below toxicity thresholds. This is one of the perks of nuclear medicine, which allows us to tailor treatment for each patient. Sounds ideal, right? However, if we look at how this is implemented into clinical practice, we find that the current reality actually looks a bit different within Europe. In a recent survey that was performed by Siegfried, we asked sites across Europe whether they perform dosimetry for treatment planning and verification of different therapies. In these graphs, you can see different therapies on the x-axis, and as you can see, for many treatments, dosimetry is never performed. One of the notable exceptions is the microcell treatment with either yttrium-90 or holmium-166, of which you will hear more in the next webinar. But for the other treatments, it was striking that dosimetry seems to have a very limited place in the therapy workflow. So apparently, many treatments are performed without carrying out any dosimetry. We know, of course, that there is a lot of debate going on about the added value of it, the costs and the resources involved, etc. But let's have a look at the risk of not performing dosimetry. If we treat as we currently do now, with for example the often used fixed dose of 7.4 gigabecquerel per cycle, we base the dosing scheme on population average risk on adverse events, and might therefore very well be treating quite conservatively. This could potentially lead to undertreatment of lesions, leading to radioresistance or recurrence of disease, which in turn would require additional follow-up treatment and therefore lead to more expensive healthcare, compared to when we would have treated right the first time. In addition, like I said, we base the risk on organ toxicity on population averages. For individual patients with higher risk on toxicity, this could lead to a decreased quality of life and also a need for extra interventions and again, more expensive healthcare. So I am not convinced that when we would carry out a proper cost-effectiveness analysis, performing dosimetry would lead to more expensive healthcare, because I think that it might actually be that investing in proper treatment during molecular radiotherapy treatment by performing it dosimetry-based could save money and improve patient quality of life down the line. Alright, so imaging-based post-therapy dosimetry. We have seen that it is currently clinically implemented to a very limited amount. So if you would like to change this, how would we tackle this? Well, the most common treatments that are currently performed are treatments for neuroendocrine tumors, metastatic prostate cancer, thyroid cancers and liver tumors. For the sake of time, I will take lutetium PSMA for treatment of prostate cancer as an example and discuss several aspects that are important to think about when you want to introduce dosimetry into your clinics. To start, you of course need to decide what are the regions of interest, because that will define the regions to scan. For lutetium PSMA therapy, the most important regions at risk are the kidneys, bone marrow and salivary and lacrimal glands, as can be seen on this PSMA PET scan. In addition, there will of course be regions of disease. So, looking at these organs, when would we actually expect toxicity? For the kidneys, the accepted threshold doses are currently defined between 21 and 40 Gray, whereas for the glands this should be below 25 Gray and for the bone marrow below 2. But you hear me say accepted threshold dose, And this is because these thresholds are often based on other molecular radiotherapies or mostly even on data from external beam radiotherapy. And we know that the radiobiological effects of these treatments can be very different, with, for example, different dose rates, fractions, energies, etc. In addition, since these treatments are often systemic, the threshold might be changed by earlier treatment and patient reserve. So when looking at the absorbed doses for the organs at risk that were found in different lutetium PSMA studies, we see that for the kidneys these are quite comparable, while for the other organs there is a lot more difference between studies. This might be caused by both the heterogeneity in the organ uptake, which would be an actual biological effect, but it can also be the study setup and the dosimetry methodology, which can be very different and yield very different outcomes, and this makes it difficult to compare absorbed doses between studies. But still, let's assume we can talk about the average absorbed dose to these organs at risk. Usually, up to 6 therapy cycles of lutetium PSMA are given of each 7.4 gigabecquerel. And if we compare the absorbed dose to the organs at risk to the accepted threshold doses in the bottom line, we see that after these six cycles we are often around or above these thresholds. So we would expect some serious toxicity, right? But do we find this in clinical practice? The answer is to a very limited amount. This is the adverse events table from the VISION phase 3 trial. And as you can see, the most found adverse events are fatigue, dry mouth and nausea. And the dry mouth, so the xerostomia, was only found to be in grade 1 to 2, which does not comply with the idea that more than 25 gray would give you serious toxicity. So this is already an indication that the organ threshold doses that we use now might be too conservative. Bone marrow toxicity was the most serious adverse event, and a little less than 4% of the adverse events led to death. And then finally, as you can see in this table, kidney toxicity is not shown, and indeed at this point it does not seem to be a serious risk in other studies as well. However, it is important to mention that especially for the kidney, late toxicity effects might be important, and little is known about that currently. So, if we want to use dosimetry in clinical decision making, we need well-established absorbed dose effect relations. Only if we have sufficient and therapy-specific knowledge on this, we will be able to adjust treatments based on these relations using dosimetry. And in order to define these relations, it is key that we standardize dosimetry protocols to make sure that the results of different sites and studies can be compared to each other. For this, we have many practical considerations, which I will discuss by taking again lutetium PSMA therapy as an example. To finish, I will provide a short overview of the guidelines that are currently available and might help you to clinically implement dosimetry at your center. For the practical considerations, many aspects have been discussed during earlier webinars in this series, including, for example, equipment calibration and quality control, and the effect of acquisition and reconstruction protocols on image quantification. As I mentioned earlier, you also need to think about what bed positions you will scan depending on what are the areas of interest, and also at what time point. Will you perform, for example, a 3D SPECT, or will you settle for a total body planar scan at some of the time points? And how many time points do you actually need? How will you calculate the absorbed dose? Will you use organ-based or voxel-based algorithms? How will you perform region drawing? And how reliable and consistent can you do this, especially for smaller structures? These are all important questions that will require a certain decision by you, and that will all have an effect on the absorbed dose that you will calculate at the end of the workflow. This again stresses the importance of the development of standardized dosimetry protocols, but at the same time it is important that you make decisions on these aspects on a local level to optimize it with the resources that you have available. I will now highlight some specifics. For example, when looking at the input imaging data, it is often tempting to choose for a planar view, because this will give you a fast total body view of the patient. However, this will of course yield a 2D view with overlapping organs, and small lesions or structures are often difficult to see. So you need to be aware that this will yield serious differences in absorbed dose outcome. This particular aspect was also analyzed in a recent SNMMI dosimetry challenge, and the outcome recommendation was that 2-day dosimetry should be avoided. This aspect was also evaluated by this modeling study by Rinscheid and colleagues, who compared a planar approach to a hybrid solution, in which a 3D SPECT was included for 1 out of 3 time points. As you can see in these two graphs, for both kidneys and lesions, the broad frequency distribution of the simulated TX based on the planar images showed about 25% of the TX deviating more than 20% from the ground truth. This percentage was reduced to below 1% by using the hybrid planar SPECT method instead. So even including one SPECT already dramatically improves your dosimetry quality, which is something you really should be aware of. Another aspect to consider is how many imaging time points you actually need for reliable dosimetry. This is especially a relevant question because we do not want to put unnecessary burden on patients and clinics, and yet we still need to be able to sample the time activity curve in a reliable way. A lot of research is currently carried out to define the minimum required time points as well as the optimal time point, as for example in this study by Brosch and colleagues, who found that a 48-hour time point for kidneys could be used as a 1 time point protocol in lutetium PSMA dosimetry. A similar result was found by a study we performed, which found a single time point between 1 to 3 days is sufficient for the organs at risk, whereas a late time point was crucial to include for the lesions, since these kinetics tend to be slower and more heterogeneous. The graph you can see is for the lesions, which shows a comparison between the 5 time point reference protocol on the x-axis and a 2 time point protocol on the y-axis, which included the 24-hour and the 168 hours, so 1 and 7 days. As you can see, it is very well possible to decrease to 2 time point protocol. And actually on this topic, a lot more work has been done already on the treatment with lutetium dotatate for neuroendocrine tumors. For example, the now quite famous Rinscheid method, which uses the information from the first therapy cycle to use in a limited time point protocol in further cycles. This is something I will not explain into detail because I will stick to the lutetium PSMA as an example here. I showed you that the VISION trial found bone marrow toxicity to be the most relevant adverse event in PSMA therapy. So this is also an important organ to define a dosimetry protocol for. However, this is also one of the most challenging forms of dosimetry. According to the EANM guideline by Hindorf and colleagues, you can either use blood sampling when no active bone uptake is expected, or quantitative imaging when it is expected. So far, for lutetium PSMA therapy, there is no active uptake expected. So, blood sampling has been the standard method for dosimetry. Looking again at what was found in previous studies, we see that we are mostly below the accepted threshold doses. Yet, like I said, the VISION trial found some serious bone marrow toxicity in some of the patients. In this study, by Grob et al., we looked into the use of imaging data for bone marrow dosimetry, comparing 7 time point blood sampling with 5 time point SPECT-CT imaging, and segmenting multiple vertebrae for the imaging-based approach. We showed that imaging-based dosimetry generally yields a higher absorbed dose estimation than blood-based dosimetry. When looking at the ratio between SPECT and blood at the different time points, we found that this ratio increases over time. This actually indicates that there might be specific bone marrow binding of PSMA, which means that using imaging data for dosimetry might be more appropriate in this disease setting. However, at the same time, this is very challenging in patients with a lot of bone metastasis, which is very common in metastasized prostate cancer. So actually, this remains a challenging aspect of clinical dosimetry in lutetium PSMA. To finalize, a quick look at some available guidelines. The EANM Dosimetry Committee has published many very useful guidelines for different types of therapies, and also some general guidelines. For example, the one at the bottom, which is an enabling guideline that gives some advice on implementing dosimetry when the resources at your center are limited. I think it is quite important to stress this one, because the limited amount of resources is often a reason to not perform any dosimetry, and this guideline actually shows you that even with limited resources you can still do quite a lot in terms of dosimetry. So I really recommend you to check this one out. There are also some joint EANM mirrored guidelines on how to perform quantitative SPECT, both in general and also for specific radionuclides as input for dosimetry. And there is also an EANM Physics Committee guideline on quantitative SPECT. While this list is definitely not complete, it can still give you a good start when you want to start performing clinical imaging-based dosimetry at your center. So I recommend you to check all these guidelines. So, to conclude, we discussed that standardization is key to establish reliable absorb-dose effect relations and enable dosimetry-based clinical decision-making. We discussed that limiting patient and clinical burden while still ensuring reliable dosimetry is important to enable large-scale clinical implementation of post-therapy dosimetry. I showed that treatment-specific considerations need to be made at a local level to successfully implement clinical dosimetry. I use an example of lutetium PSMA therapy for this, but all these aspects also hold for the other therapies that I show you. And finally, I showed that there is quite some guidance documents available already, and I also want to mention here that there is some of them that are now in preparation and many more are following. Many committees are now working on helping us to perform dosimetry in a standardized way.